Welcome to the Food Tech. In today's video, we are going to discuss bacterial reproduction. For detailed notes on this topic, check the links in the description. Let's dive into the microscopic world of bacterial reproduction. The most common way bacteria reproduce is through binary fission. Imagine one bacterium splitting into two identical copies. This process is rapid and highly efficient. Let's break it down into simple steps. Step 1. DNA replication. The bacterium first makes an exact copy of its DNA. Think of it like photocopying a document before sharing it. Step 2. Cell elongation. The bacterial cell grows, stretching itself to prepare for division. Step 3. Septum formation. A new dividing wall called a septum starts forming in the middle. Step 4. Division. The cell finally splits into two identical daughter cells, each containing a copy of the original DNA. Under ideal conditions, some bacteria, like Escherichia coli, can divide every 20 minutes. That means in just 7 hours, a single bacterium can multiply into over 2 million bacteria. That's why bacterial infections can spread so quickly. Unlike humans and animals, bacteria don't reproduce sexually. But they do have methods to exchange genetic material, allowing them to gain new traits, such as antibiotic resistance. Let's explore 3 fascinating ways bacteria do this. First one is transformation. Some bacteria can pick up free pieces of DNA from their surroundings and incorporate them into their own genome. It's like finding a useful piece of code and adding it to your system. Second one is transduction. Here, bacteria get new genetic material through viruses called bacteriophages. These tiny viruses infect one bacterium, pick up some of its DNA, and then transfer it to another bacterium. Imagine a USB drive that accidentally transfers extra files when plugged into different computers. Third one is conjugation. This is the most direct method, where two bacteria physically connect using a tube-like structure called a pilus. Through this bridge, one bacterium transfers a small piece of DNA, often carrying important traits like antibiotic resistance. This is why some bacteria become superbugs that are hard to treat. Did you know that scientists first discovered transformation in 1928 through a famous experiment by Frederick Griffith? His study on pneumonia bacteria led to the discovery of DNA as the molecule of inheritance. Bacteria are survivors. When faced with harsh conditions like extreme heat, radiation, or lack of nutrients, some bacteria form tough, dormant structures called endospores. These spores can remain inactive for years and reactivate when conditions improve. Did you know? In 2000, scientists revived a bacterial spore from a 250 million-year-old salt crystal. That's older than dinosaurs. Understanding bacterial reproduction is crucial in microbiology, medicine, and food safety. It helps us develop antibiotics, control infections, and even preserve food better. If you enjoyed this video and want to learn more about microbiology and food science, hit the like button, subscribe to the Food Tech, and turn on notifications so you don't miss our future videos.